professor at Kiev University, harnessing biotechnology to monitor, simulate, and investigate cellular activities of living and microorganisms such as fermentation. Dr. Zubi aims to pioneer integrated systems biology with information technology. As another note, within 3 minutes of each speaker, you will hear this noise, so please do not be alarmed.
Hello. Thank you. Thank you for your introduction. Hello. I'm glad to see you all. Thank you very much. I am Haruo Suzuki from Keio University. So my talk title is MetaSUB and Japan. MetaSUB と日本。Let me check how to use this. Pointer to forward back. Okay. So here I listed keywords. There are many keywords. Probably I will not cover all. Keio, SFC, MetaSUB, Japan, Olympion, time variation, batch effect, AMR, plasma, COVID-19. I think these keywords are important in this project. And firstly, Keio University, SFC. So I am from Kanagawa. I'm living in Kanagawa, next to the Tokyo. Kanagawa, here, Tokyo here. And you might notice, you might watch the TV news. Earthquake hit near Tokyo, actually, Western Kanagawa Prefecture, 4 days ago. And my university is located in this Kanagawa Prefecture. This is my campus, Keio University, Shonan Research Campus, SFC. And this is one of the sampling location we have been working on. And we also collected samples nearby station called Shonan Station. So I have been collecting samples from Shonan Station monthly, every month, since 2021. Guess who he is? Okay, a second. So his name is Yukichi Fukuzawa. He's founder of Keio University. And also, he's the most famous man in Japan. Because maybe you have already heard this banknote in your wallet. So everyone prefer him, I mean, this banknote. And this is his statue, bronze statue. Of course, we have already collected samples from him. Yeah, for as a part of monumental project, and also collect samples from banknotes to test whether this surface can be used as a platform for monitoring the status of the global microbiome. So in this university, I have been teaching bioinformatics using this textbook and sequence data analysis using RStudio and other open source software. Okay, let me quickly introduce one of our work related to built environment. We published this paper in 2019 in DMC Genomics. First author, Dr. Nancy May, she surveyed 60 papers related to built environment. In this paper, the author researchers investigated microbiome built environment using shotgun metagenomic sequencing data, 16 ribosomal RNA gene amplicon sequence data, and culture-based method, culturing method. And we split the publicly available complete genome data into 2 groups. Excuse me. is split into the complete genome into two groups. Bacteria, commonly identified in the built environment, and other genomes. And we compare genetic features, genome size, GC scale, GC content, S value. S value is the degree strength of selective codon usage bias. So this statistic can be used to measure growth rate of the bacteria. And we found that bacteria in general, commonly identified in built environment, tend to have higher S value compared to other genomes, suggesting that. So this research generated hypothesis that bacteria in built environment tend to have higher growth rate, grow faster compared to other bacteria in other environment. So yeah, we are very interested on the bacteria in built environment, including school, hospital, and subway and bus stop. Yeah, public transportation system. And MetaSUB. Yeah, this is the paper published in 2021 in Cell paper by David Tang et al. And MetaSUB Consortium. And this is a paper, an article published in iScience in 2022 by Christian et al. MetaSUB Consortium. So here, the article title is A History of MetaSUB Consortium. And yeah, please take a look at this article. So there are history, and you can, you know, some spin-off project of this MetaSUB is very fun. Very interesting article, I think. Okay, so let me talk about data and metadata I submitted in public database. I created umbrella project for MetaSUB to collect all the data and the metadata related to the MetaSUB project. And if you have internet, laptop, please access this website, NCDR. And the bioproject accession number is PRJDB13767. Actually, if you type MetaSUB in search box, and probably you can reach this page. So hopefully I would like to collect all the MetaSUB related data in this website, so anyone can access MetaSUB related project data, including PASMAP 2015 data, and meta-air data, and the global sea sampling data published in CERN. And also I created a backup project for metagenomic samples collected in Japan. Yeah, this is the example of microbiomes in Japan bio project. Excuse me. Okay, so Japan. In Japan, we joined global city sampling year at the beginning in 2016. So we swapped many places, many surfaces in Japan. So here is a summary sampling effort in Japan. So we joined the global city sampling day in 2016, and we conducted the first winter sampling in 2018. And of course, we joined the Monumental project. We also, in Japan, as a unique project in Japan, we collect samples before, during, after the Rugby World Cup in 2019, one of the mass gathering event project. And after that, we joined the MetaCoV project, RNA COVID-19 project. We joined the MetaCoV project since 2021. And in 2021, we conducted samples before, during, after the Olympion and Olympics, Paralympics as a part of Olympion project. This is the sample map in 2021. If we focus on Japan, so in this year we collected samples from many cities across 15 prefectures in Japan, as shown here, from Hokkaido to Kyushu. So these are statistics, number of samples, tubes collected in Japan in different year, months, and different place. And I sum up some of the number of samples collected so far. So this plot shows number of samples collected at each time point. So of course, in June, globally sampling rate, the highest number of samples were collected. But we also collect samples every month in some city, including including near Tokyo, Kanagawa. So this plot shows the number of samples collected at each prefecture. The highest number of samples were collected in Tokyo, followed by Niigata, organized by Dr. Abe at the Niigata University, followed by Kanagawa. We collected samples, followed by Hiroshima. Dr. Mariyama organized sampling in Hiroshima City, followed by Chiba. So here is a summary. Japan, the project in Japan. With collaboration CREST project and the MetaSUB consortium. For sample collection, over 3,700 samples were collected in 50 cities across 21 prefectures in Japan in 2021 to 2024. Sample processing in Japan. So of the 3,700 samples, most samples were sent to Kone, New York City. The remaining selected samples were subject to sample processing in Japan. So far, we performed shotgun sequencing of approximately 488 samples. So we have 488 metagenic samples in Japan. And these data can be accessible in the public database in CDN. These are number of metagenic samples available for each fiscal year. And using these data, we perform the data analysis, including taxonomic assignment, functional adaptation, antimicrobial resistance, and NAG, metagenome single genome, and mobile genetic element, including platelet. Okay, let me briefly explain our result. This shows the number of daily confirmed cases of COVID-19. So you can see the wave, different wave of the COVID-19 pandemic. First, second, third, fourth, fifth, sixth, seventh wave. Now it's 11th wave of COVID-19 pandemic. So, fifth wave in 2021. Okay, so we collected samples from Narita City, Chiba Prefecture in August to September in 2021. So this time frame corresponds to the fifth wave of COVID-19 pandemic. So we would like to see the normal microbiome during the pandemic, fifth wave. We found many bacteria commonly identified in rich environment, including apne and other bacteria. And in the fifth wave of COVID-19 pandemic, we also collected samples from university campuses, 3 university campuses, 3 surfaces, table, sheet, and floor. And we found interesting result. We found some known pathogens like Acinetobacter and Staphylococcus. So further study will be required, but should be interesting. I'll let you know the update later, someday. Okay, so this is another result. So we performed here comparative metagenomic samples. We compared samples collected in Shinjuku Station in Tokyo and Niigata Station in Niigata Prefecture. So here are metadata, sample name, DNB-seq. We performed shotgun sequence using DNB-seq. So sample name is DNB-seq. Number, these are collection data for the samples from 2021 November to 2022 June 21st to global seed sampling day. Yeah, in Niigata Station and Shinjuku Station in Tokyo, we monthly collected samples. When we compare these data, we found that temporal variation within a city are smaller than variation between cities. So the results are similar to those in the cell data using the 5,000 measuring samples. I forgot to explain this heat map. So this is a clustering result based on taxonomic composition inferred using MetaSUB for software. Rows are samples, DNB set, sample name. Columns are species. So you can see heat map. So red indicates higher abundance, blue dark color, black indicates lower abundance. So acne, C. acne, higher abundance, and other oral bacteria are also skin bacteria, microbiota are higher abundance, as expected. And in this clustering result, Niigata samples clustered together, Niigata, and Tokyo sample clustered together here. So suggesting that seasonal variation within a city tend to be smaller than variation between cities. Yeah, this is interesting. And using all the data available, we can conduct a detailed result to test this hypothesis. So we have city-specific microwave sewage. And these results are presented, reported in the International Symposium on Data Science 2023. I reported this title, Urban Microbiome Monitoring in Japan, collaborative research with MetaSUB Consortium. And Dr. Daly gave a talk regarding a comparison of microbial diversity and resistance determinants in the field environment in Japan using shotgun metagenomic sequencing. Dr. Daly, she also conducted research using culture-based method because antimicrobial resistance gene identification only based on sequence data is not enough because even bacteria have resistance gene. Sometimes they do not have resistance to the antibiotics. Sometimes they are very sensitive to the antibiotics. So she is studying this title, multi-drug resistant staphylococcus captured from urban public areas, a culture-based approach. Chris and I are from co-authors. I briefly explained the Japan project in Japan. In lightning talk, other Japan members will provide reports. So let me briefly introduce these presentations. So firstly, Dr. Takashi Abe at Niigata University, he will give a talk titled Phylogenetic Analysis of Urban Flora in Niigata City, Japan, using bat learning self-organizing map SOM based on the similarity of K-map compositions. MetaSUB and other software assign taxonomy, analyze taxonomic composition based on alignment, right? Sequence alignment. So instead, Dr. Abe used K-map, oligonucleotide profiles, for taxonomic composition analysis. And Dr. Yu Shiwa, from Tokyo University of Agriculture, he analyzed RNA meta-transcriptome. So his talk title is Evaluation of Ribosomal RNA depletion methods for capturing the RNA biome from environmental surfaces. So we know the concentration of virus on surfaces very low concentration. So we need to improve detection of these low abundance viruses. So Dr. Shi has been studying this topic. And Dr. Kirill Kurikov, he will talk about using genome search toolkit, GSTK, for the analysis of short-term metasome sequencing of samples from Japanese urban environments. So he applied his pipeline for the analysis of short-term sequence data, metagenomic samples obtained in Japan. Yeah. Dr. Honda at Canada University, he has been studying the antimicrobial resistance in wastewater, sewage, and he joined the MetaSUB project 3 years ago, and he studied this topic, metagenomic profiling of antimicrobial resistance insights from the MetaSUB project data from Japan. Yeah, please stop by this writing talk. Let me introduce this specialist. So, the microorganism journal, we have specialists, the urban microbiome, best fitting topic. And Dr. Sujin and me, best editors. Deadline extended to October 15th. So, please consider to submit your result. This is acknowledgement, but I still have some time. So maybe, what should I stop here? 15 minutes. Okay. There are different topics I would like to mention. Okay, plasmid. So let me talk about the mobile genetic element, plasmid. Plasmid is a extra chromosomal element. So bacteria, chromosome, plasmid, small DNA. The plasmid often carry resistance genes, antimicrobial resistance genes. Plasmid carry AMR genes. The plasmid can transfer between species, genera, or sometimes between phyla. So plasmid can be broad host range and narrow host range. If multi-drug resistance genes encoded on broad host range plasmid, this is a big problem because these resistance genes spread diverse bacteria in the environment. So we have been studying plasmid in collaboration with National Institute of Infectious Diseases, Shizuoka University, and the University of Tokyo, by supported by OSMI, Frontier Science Foundation. This is one of the results of our analysis. So we found that strong correlation between plasmid GC content and their host chromosomal content. Very strong GC content correlation. So we can use this information to predict plasmid host range. So we have been developing this method using GC content and oligonucleotide composition, KMA profiles to predict broad host range plasmid and narrow host range plasmid. And now we work on developing database of plasmid for taxonomic assignment of plasmid mobile genetic element in metagenic samples. So this is one of the paper we published in 2022. Precise classification of antimicrobial resistance associated increased metaplasmid. So, there are many misannotation error in public database and paper. So we correct these misannotations, and we also published paper entitled Reconsideration of the previously classified incompatibility group plasmid, INC-P1 and INC-P2. The name of plasmid, naming scheme of plasmid is very difficult to understand. So now we manually created and creating our own database. So as shown here, there are incompatibility group plasmid. When I first started studying plasmid, it's very headache to understand these classification like ink P1 plasmid group. Ink P1 plasmid equal ink P. Ink GU plasmid equal ink P6. It's very different. Usually people think ink P1 and ink P6 sounds like subgroup of ink P group, but this is not correct. So this is very complicated. So now we manually created database and creating our own database. And hopefully, we would like to report the result before the International Symposium on Plasma Biology 2024, which will be held in Kamomachi City, Japan, September 2nd to 6th. Yeah, now we are preparing an article. So I would like to use this database to annotate mobile genetic element in MetaSUB metagenomic samples. COVID-19, maybe I should talk about this person. So, as you probably received email from Krista, and you saw this case, number of patients in Japan. So now we are in the middle of COVID-19 pandemic 11 sites. So yeah, please be careful. Virus everywhere, microbes everywhere. So it is recommended to wear masks if possible. I have this device today also, but I use this device called RNet for MetaSUB sampling to collect metadata for MetaSUB sampling. So this device is very good, great tool. It is recommended. I tested different device to measure carbon dioxide, CO2. So you can, using this tool, you can monitor CO2 concentration every minute. So I think this is very useful for metadata collection in MetaSUB project. And please consider to buy for your project. Okay, mass gathering event. And this is the future plan. So we conducted samples in Rugby World Cup and Olympics, and now probably Paris Olympics just ended, right? So Olympian in Paris just ended. It is very interesting to compare these different metagenic sequences collected from Rio Olympic 2016, Tokyo Olympic Paralympic 2021, and Paris Olympic in 2024. And we have more mass gathering event. So people bring their migrants with them. So we will planning sample collection before, during, after the mass gathering event in 2025, 2026, 2027. So I will report the result another time. Okay, and finally, metadata. This year I asked people participate in the sampling. Please record what you ate before your sampling, because your food might be detected in your sample. Okay, excuse me, that's it. Thank you very much. Sorry. That's it. That's it. Sorry.
That's a 3-minute timer, so we have 3 minutes before Q&A.
I think shorts. Supported by this grant. Okay, thank you very much.
We'll take any questions.
Dr. Suzuki, that's a good compilation of what is going on in Japan. I have a few questions. You have shown a nice COVID, patterns since 2020, but is there any data before 2020 you know,
Yes.
is that you can, find this, COVID signature before, March 2020.
That's interesting.
Do you
That's
have,
interesting. Yeah.
do you have?
Yeah, we have data. I mean, and the data collected in 2018, 19
Yeah.
will be available soon.
So you have not checked it?
I haven't checked.
Okay, so who are interested there, maybe good to know whether, any kind of those signatures found before the outbreak. Okay, so I have a few other questions, okay? You have shown variation within Japan cities, but have you compiled your data and compared with other countries?
No.
No.
This is because, I didn't talk today, but budget is. It is very difficult to compare data collected from sequencing different time. So sequencing run does matter. So this is a paper from microbiome published in 2019 or 18, sobre microbiome. Seasonal dynamics and direct comparison of air and surface microorganisms. So they reported that sequence run was also significant predictable among sample diversity, data diversity, and explained 2% of the variance. Ordination plot, TCOA plot revealed clear differentiation of air surface, season, and sequencing run.
So basically, with the methodological difference, it is not possible to compare your data or anybody's data with other city?
I wouldn't say impossible, but need some batch effect correction. There are various
Always batch effects.
complications.
Always batch effects. If you're asking me to sample myself or Nikos, we will have a different way of collecting samples that is there. What do you think, Chris, about that? Chris, we cannot compare one's data with others because of the methodological problem?
We haven't. We haven't done yet.
Okay.
But shall we try?
That is, I think, one of the major objective of the global MetaSUB. Otherwise, there's no. So along the same line, now you collected samples in Tokyo as well as Niigata, okay? And you have clearly showed there is a difference between those cities. Is that because the Tokyo is a cosmopolitan city, you have so many different people comes and go, or what?
I have no idea. We need further research. We need more metadata to. Maybe, but we haven't done it.
Maybe the Shinjuku station might have millions of people going in and come, but Niigata is not. That's one way. The other way is the influence of the Pacific versus the Japan Sea. Maybe the other one. Okay.
And Niigata, in winter, Niigata have a lot of snow. So maybe these climate can influence the microbiome.
So one last thing, you have the surface sample collected, but I'm seeing the floor only. Why don't you get these samples from the ticket vending machines or the escalators, the rim?
In some cities, we collected samples from these surfaces, but
this is floor, right? All these things that you're
this is
talking about.
floor,
Floor means which floor? Where did we collect?
like floor.
No, this is the station, the train station, the floor only, right?
In front of police box, police station.
Yeah. So that means, mostly the soil microbiome you are collecting rather than human transmitting one to the other. Maybe it is better to also collect some samples from the ticket vending machine and also the rails that people touch, right? So there you may have a more different kind of microbiome than the floor. That's my thinking.
I understand the sample suggestion. We collect samples floor because previous results reported that floors or ground show highest alpha diversity. Like Suzu's result in Korea station, yeah. And other built environment microbiome data study reported that floras of the different surface type, floras show highest microbiome diversity. That's why we select the flora as a representative surface to monitor our microbiome. But yeah, of course, your suggestion is very appreciated. Thank you. Thank you very much.
Great, thank you. We have time for 2 more questions.
Hi, everyone. Great work. You've been really doing a lot. The plasma data set, that correlation in GC content, and that would be expected genetically based on essentially close range adaptation. So I'm curious, have you looked at parsing your plasmid data set? So you're not looking at the structural genes, but rather on perhaps newly acquired elements on these plasmids to look and see how just date to some extent artificially the amount of time since acquisition of some of these resistance elements in the plasmids.
Sorry.
So if you separate, when you assemble your plasmids, if you separated the data so that you can distinguish between your core rep elements and, say, newly acquired antimicrobial resistance genes to try and identify ICEs and other mobile genetic elements within the plasmids themselves, if you could see some deviation from this GC correlation that you see here.
I investigated this and checked different region of blood, core region, accessory, when I worked in a lot of stuff at the University of Icon. Yeah, we got a very unexpected result. Yeah, I don't know why, but the accessory region has more strong signature between plasmid and host than core gene. So we expect core gene should have strong signature between plasmid and host, but our result contradictory this. So we have.
Awesome. We'll talk about it later. Great.
Yeah,
Thank you.
then thank you. Remind me this.
Thank you, Aro. It was really great talk, and then I can see you're very actively working on a lot of projects. So one thing is that kind of similar to the what Patrick asked you regarding to the 2 different cities, the microbiome differences. What I found in our studies that in Korea we look at the very small region in Seoul, but when we look at the microbiome differences between the subway station versus the bus stop, what we found is there is differences, but the differences comes from the environment. So if we look at only the microbes likely originated from the human, there are not much differences, but the environment itself is kind of contribute to lots of difference between 2 sampling sites. So I wonder whether your study actually Nigata as well as the Tokyo, maybe the 2 different location of the city kind of the environmental microbiome contribute to your differences instead of the human originated microbe. Have you actually distinguished these 2 different microgroups?
No, we haven't done it, but yeah, we should check. Maybe we can use source truck or something.
So the way we did was we only look at the microbes which based on the known database like in the human skin microbiome and then the gum microbiome, things like that. So it's not perfect, but we can actually see what's likely comes from the human versus environment like a soil or water.
Did you use some specific software?
No, it's at that time we just almost like a hand, manually we did it because we can see the core, the microbiome species. And then when we look at it, we found it is.
Yeah, basically, maybe we can use micro directory dictionary for this purpose.
Maybe because I remember the cell paper, the consortium paper, we compare the entire data with the skin microbiome and soil microbiome. So if you apply similar ones, then probably you can see whether the difference comes from the environmental microbes or the human organism.
In the case of Korea, you collect the samples outside or inside or mixed place.
Actually, so bus stops is outside, so it's open to the environment, really open to the environment. But the subway station is on the ground, so it's more like very contained places.
And more human.
Right, exactly. That's what we found.
Yeah, thank you for your comment. Yeah, thank you very much.
Okay, thank you. At this time, we'd like to invite our next speaker, Dr. Levi will be presenting.
So you can sanitize this. Yeah, sanitize using the mic.
Oh, please use the sanitation at the podium. Thank you. And Dr. Levi will be presenting on targeted sequencing and triplet loss classification allowed for microbiome-based interference of soil sample origin. Dr. Levi is a bioinformatics group leader and professor at Jagiellonian University, and he is at the Malabowska Center of Biotechnology, Jagiellonian University in Krakow, and the European Continental Director for the MetaSUB Consortium. His work is on bioinformatics pipelines for data science, focusing on transcriptomics and metagenomics for precision medicine. Thank you, Dr. Levi.
Thank you. So good morning, and it's great to be here. I would like to welcome everybody, and I hope you will enjoy. So this talk is not directly about the MetaSUB related project, but it was inspired by MetaSUB. And as Chris was saying, all young parents, okay, it's not working, are considering whether they're these goods, right? And it was also one of the things what I was thinking about when I was still younger parent. Later I will start using whether the dirt can be useful, right? So in the first study it was shown that first of all, the most of the research, we are reading, they are coming from unknown organisms, and in different cities it in fact is going more than in New York. And then you can see the different compositions in different parts of the cities, in between the cities. So we start wondering how we can go further. As it was already shown by her work, there is a paper where you can read about the beginning of the MetaSUB. I started MetaSUB in 2017 or 16 in Vienna. Then when I moved to Krakow, I started it also there. Last year we, following what Paro was doing, we also had the European Games, so we're collecting the samples before European Games, during and after. We are now the sequencing part of the data and analyzing them. But it was bringing, such an event was bringing an idea what can be else done. Within here with the consortium, we have different backgrounds, so we are looking at those data from different perspectives, which is great, that we bring all those different people together. But also to open it up, already at the beginning, we started donating, let's say, the MetaSUB data into the Canada. There is a type of this critical assessment of massive data analysis. So this is the conference which I'm running as a part of ISMB, and which is trying opening up the big data to the community of data scientists mostly. And we are giving the data with open-ended question, asking, can you tell me something new, interesting about those data? And we started, I think in 2016, Abraham was showing up what the MetaSUB is about. And then from 2017, we started giving more and more data into the MetaSUB. Come back community, asking people, can you distinguish between cities? Can you classify from where the sample is coming from? Or can you predict the origin if it's coming from the city which is not in the learning data set? So this was the questions which we were keep asking during the campaign. And the outcome was that there is a city-specific microbiome profiles, which allows for the classification with a high accuracy, and it doesn't matter whether you just following the classical taxonomical characterization and using this as a features, or you prefer going a bit further, that looking into all different biomes in order to try to focus on the functional profiles and then classify using those features, or maybe you don't care. You're just simply using this as a bag of words, and you don't care really what feature it is, because the characteristics of the data set is the same, just looking to the k-maps. In all of them, it was working very good for the classification, but on the global scale, the microbiome composition multilateration is not working. So we cannot really predict the origin, at least not with the resolution of the data we still have in the MetaSUB. So we had some odd classification like the New York and Lisbon, but you can see that they are, bold at the Atlantic Sea on the both coasts and more or less the same distance from the equator. And the second part of the misclassification, it was always a so-called trouble for the people who were Tokyo and Auckland. And again, we see the same distance to the equator, the Pacific Ocean. What Benta was saying is that those, like in Japan, those cities have completely different populations. And the same is here. In New York and Lisbon, it's order of magnitude. Tokyo and Auckland, I could probably say even 2 orders of magnitude. But still, from the microbiome compositions, they cause problems, and they cause misclassification. And it was really, doesn't matter, it was a common problem, it doesn't matter how people were looking for those data. So there is something unique which is coming from the environment, which is dominating through the other factors. But those misclassifications make us aware that when we look into the Earth, continents, oceans, so this is what defines, and with the climate, some kind of ecological niche, which is this defining what the specific biome is, and that's why the multilateration will not work, because the biomes, they do not change linearly. So if we have 3-4 points, we cannot really predict what's in the middle. But it was, our take-home message from Kamla, more or less at the same time, because those were years 2017, 2020, the analysis with the MetaSUB was ongoing, which has ended up with this nice paper, which also asserted that urban microbiomes are important features of cities and city life, but also the environment. It was also shown that we can really nicely predict with a very simple classifier, unremade, really great tool, about saying from where the specific reads are coming from, and then as the follow-up work by Chandrima was investigating different approaches for the machine learning perspective, and in fact was showing that yes, it is possible to classify cities and continents, and really when you look into the continents, it's easier. Cities is more difficult. In principle, all combinations work good, but there are some which are working better, right? But then you can start thinking, okay, this is globally, so it should be easy. But what if we will look into, more homogeneous area? So once I came to Krakow in 2017-18, I've met the people who are working in the forensics. We started collaboration with the Central Forensic Laboratory of the police, and we started wondering, okay, can we go with what we see in the MetaSUB a bit further? Can we try to understand, and can we apply the knowledge we learn in Canada and MetaSUB for forensic investigations? Whether that is to check whether it's possible. Because the microbiome forensic is something which is pretty well described in different applications. I mean, it's already pretty easy and very for many years that you can think looking into which bacteria are there, you can decide, you can estimate what was the time of death. From the work of our colleague from Hong Kong, Patrick Lee, It's also understandable that you can identify the human microbiome, and by this to identify whether a specific person was in this room, even a few weeks after. But what was not investigated before, it was like the surface or soil microbiome, whether you can use this for forensic investigations, like whether we see that there is a trace and it's clear that this dirt is not from the crime scene, can we analyze it and then tell from where the sample is coming from? I mean, people can say, okay, you can do the physical chemical analysis, check the soil type and so on, but then you need a lot of material for this. And this is not what in many cases you have in the forensic applications. So the microbiome might be an answer, and this is what we wanted to investigate. So this was the applied project in Poland, which was called Soil Microbiome Analysis Forensic Tool. And we had to be very careful how we define what we will deliver, because it had to be applied in a way that we had to provide to the police the ready-to-go tool, which they can use out of the box. So this was a collaborative work of the Leiden University, the Central Forensic Laboratory of the Police, Pomeranian Medical University, Warsaw Medical University, and the company Arvigen. So the concept was that it's clear, of course, in MetaSUB we sequencing 5, 10, 20 million reads. We would like to sequence deeper, but this is not what typically the forensic lab will do. Other thing is that we sequence in batches, hundreds, thousands of samples. This is not what will be useful for the police. They want to sequence, they want to have an answer. They cannot wait till 1,000 samples will come from the whole country. So the idea was whether we can do the deep sequencing of the soil microbiome to design the panel for targeted sequencing, and then this panel could be used in any lab because it will require just 500,000, 1 million of reads. So the first thing was, okay, the Poland is stopped working. Yeah. Okay. We have press something. Maybe not. No. Okay, now I will just use this one. So the bullet is pretty round. I mean, it's defined depending how you count the third or fifth, the most round country over the world. In principle, it's flat, not like Japan that you have the mountains everywhere. We only have some mountains on the south, but in principle, it's flat. So it's very homogeneous in terms of the climate. Still, we wanted to pick set of the points, so we wanted to see whether there are any sub-climate zones in Poland. So we analyzed the weather conditions for the past 20 years to just select them, and we managed to select them. Later it appeared that this is connected with the vegetation zones in Poland, so it was a pretty cool discovery. So this is what we did, that we just selected those zones, and then we defined 80 locations representing clusters spread around Poland. In each location, we collected three samples over 4 seasons, so it have almost 1,000 soil samples, which were sequenced pretty deep, over 100 million reads per site. So together it's over 12 terabytes of the data. Plus tons of the metadata collected with the Cobalt toolbox. We also did the physical chemical analysis of soil samples collected in spring, and we have this open weather map data for 20 years. And the idea was that to treat everything as a bag of words, because for forensics we don't care about taxonomy, and knowing that 40, 50, 60% of the reads they do not classify to anything what is known, and that the databases are, they keep updating, we cannot just, and you have recently the redefinition of the taxonomy, so we cannot rely with the prediction for the police on this. So the solution was that we focus on the camera-based approach, the bag of words, and we just want to do the proper classification. So we use the MetaGraph solution developed by by Andrea. And the concept was that we took the MetaSUB data as a negative, set of the features, our smart data as positive. We run the MetaSUB, we define the unitics, so the stretches of the sequences which are unique for the specific location. I put the asterisk with the unitics because after the first part, we identified about a few millions of unitics which were informative for the distinction between the locations, but of course we cannot put the few million probes on the panel. We wanted to go to be as small as possible, as cheap as possible. So then we said we ran a few rounds of the feature selections using the information gaming theory based MDFS and Boruta to get about 1,000 unitics, which were there used to design the panel and get reference data. We tested a few types of the panel. We tested two amplicon-based, but it appeared that we have not predicted that we might need a couple rounds of designing the primers for the amplicons, testing them, validating, and then some of them might not work, and then just we need to run more. That's why in the end we decided to go for the probe-based, because the probe-based was working very well. And the interesting thing was, we were expecting this, but it was very interesting for our colleagues from the forensic lab, that from about 1,000 sequences, so this unitics, over 40% are coming from unclassified, so they are from not known yet organisms, which is good for forensic. I mean, if you don't know what is there, you don't know how to falsify the trace. And looking into the typical analysis, we then can see like the dominant feature is taking like the 4% of the signal, top 10 is about 15, top 30, 40, so it's already, so there is already something which is dominating here. And you can see also here that those unclassified, they are pretty dense in the top 30, which is again a good sign for the. One challenge which we wanted to do is really whether we can make this transition from the deep whole metagenomic sequencing to the targeted sequencing. And you can see here on the y-axis, on our x-axis, you have whole metagenome sequencing TPM values. On the y-axis, you have TPM values for the targeted sequencing with about 1 million reads. And it was with the Roche panel with the 5 nanograms of the starting material. You can see typically it's like the upper one, the worst which we are getting are the switches on the bottom. And it doesn't matter really which metric you are using for similarity assessment, it looks really good. This is just whether we are getting the same signal, because the concept was that we build the model on the whole metagenome sequencing, but we do the testing on the targeted one. And then the classification was that we are getting the new sample, we have our panel sequences, you're doing the quantification, and we're using the triplet loss based approach to get the prediction, and the predictions appear to work very well. I mean, there you have different metrics, you have 4 bars, each bar is a different season, so you see that it's working pretty nice. You see that there is a drop in green, which is spring, and it was a bit surprising for us, because one thing which we learned was that the tools which normally are designed for the analysis of data, like 5 to 10 million reads, they work very bad in terms of the computing time when we have more than 100. So in principle, we were overloaded all the computing clusters in Krakow and nearby, which made that till our deadline, we were not able to process all the data. So we based our panel design on the 3 seasons. So the fall, winter, and spring, and the summer was not really included into the process of design, but it then was there for testing. So our thinking was that the worst result would be for summer, but it's not, it's for spring. But then when you go back and start thinking more, you say, okay, this makes sense. The spring is time when everything is bringing to the life back. So there's the biggest change, the biggest dynamics happening. So it makes sense. You see that it's working. And because it has to go to the forensic, to the lab, they say, okay, you cannot just do, shiny, nice lab testing, right? You need to do some kind of the proof that it will work also for the typical use cases. So the upper one is the validation set, so the lab work, typical work, and here we have the mock forensic set. So what was the mock forensic set? The soil was put on the jeans and on the sole of the shoes. Then it was stored in either the fridge, room temperature, or in the warm place for 1 week, 1 month, and 3 months. And after this time, it was collected, and then we were doing the sequencing and all the analysis. So you see that the drop is really marginal for this mock forensic case, which was very good in our project. So in summary, the big data, we have the deep whole genome solid microbiome sequencing. I mean, as far as we know, this is the unique data set. There's no similar data set anywhere all over the world. We use the metagraph because in the forensic we don't care which bacteria are there. I mean, they need to be informative. And we use the Baruta-based relevant feature selection. We use the pro-based targeted by Roche metagenomic sequencing, and 1 million per sample then is enough, which makes it can be used by any forensic lab using either the MetaSUB, Illumina, or S5 with the Thermo Fisher. So it performed extremely good during validation and pure science. So sample participants per sol performed not worse for the mock forensic. We developed and delivered to the Ministry of Internal Affairs the full system. So at the moment it's at the disposal of the Polish police. They can use it. Whether they will, it's their choice. And I think what is interesting for future is, as this approach works in as homogeneous country as Poland, it will work everywhere. So this will be the end of this part, but I just wanted to say that we do not stop here, because those data are so cool, so big that we want to follow up. I mean, Alina is here, can say a few words about this. Daria has a poster, and Rodolfo is going to talk about investigating further those data on Thursday. But in principle, we have so much interesting data that we would like to go further. So currently we are looking both in taxonomical and functional interpretation. We check influence of weather conditions, also the physical-chemical soil composition, looking into the AMR, viruses, phages, MAX, fungi-based analysis. So we are now investigating all of those. If you think that your approach or your tools, your whatever you're doing might be also interesting to check on our data, just pop by to me, Daga, Rodolfo, or Alina, and just talk with us. And the second thing is, once we will sort this out, this characterization, next step is that this is so deep, and you see that we see new species, that even from metagenomics we'll be able to identify new species which are coming from Poland. So this is the plan probably for the next year. And one more thing is that now there is an open call for the Horizon, so European funding framework, which is open topic in the civilian security. And it's under the open topic, the proposals are welcome to address new, upcoming, or unforeseen challenges, and or creative or disruptive forensic solutions for fighting crime and terrorism. So if you would be interested, I mean, we need to check which countries are eligible, because I don't remember at the moment. The application deadline is November, so we need to form the consortium now. We have set up the ideas what to do. There is a reasonable budget. They expect the technology readiness level 5 to 7, yeah? So between validating a relevant environment till system prototype demonstration, something in between. So it's not fully applied like ready system, it's not that advanced, but it's not just basic science. Okay, thank you for your attention. The work was funded by a new generation of the data was the National Center for Research and Development. The follow-up work is going through the Sonata Beast work. We especially want to thank the Cifrones, so the High Computing Center. In fact, my student got the newest high computing facility 2 years ago for its own for 3 weeks before it was officially open for others, because otherwise we will not manage to do all the calculations. So thank you for your attention. Happy to answer the questions now or during the break.
We have about 5 minutes for questions.
thank you, Paul, for a very nice new analysis. Hope that we can get something. So you said that around 48% are unclassified, but do you also see anything that you will throw the sequence of your deep sequencing, in the sense any microbial dark matter? Because in my case, for clean rooms, we are throwing almost 60-70% of the thing that is not even coming to any root. So this unclassified 48% you said, is that also included, or you just throw those?
I mean, those over 40% which we have unclassified, those are the feature types. It means that we have this a few levels how we select them. So they have to be informative in terms of the distinction between different locations. So it's not that something just by random seeing in one sample and we take it, right? It has to be the specific location on the reasonable level of the abundance, because as we are using the probe-based targeted sequencing, we are not doing the antigens, right? We're just doing the probe-based amplification, which means that we're just shifting the abundance by 2 to 3 orders of magnitude.
I'll talk to you more detail because there's different question I had, but
Exactly.
one interesting thing that you have told about, why we could not differentiate microbiome composition between the globally, okay? Is it because of the dominant microbes that are, human associated? Is there any way that one can deplete those dominant microbes and look for the one that is, less than 1% reach, that may be different between Lisbon and New York, for example?
I mean, this claim was mostly that at the moment we don't have the high resolution of the place from where we get the samples. I mean, once we will have, data every, let's say, 50 kilometers network, I don't think we ever have it, but if you would have it, then it would be possible. I mean, the thing is that when, I guess this is the word like around this link, when you're doing the ancestry tracing, you can just pretty nicely get information from where the specific persons are coming, the family, everything. With microbes, you cannot do this because even the mountain chain is changing within 100 kilometers, changing completely the biome. So it's not linear there. So I think the resolution is at the moment too low.
The last question is your panel design. Is it based on dominant microbes?
Sorry, the panel?
The panel design, is it based on dominant microbes only?
So the panel is based on the features which we have selected. So the question, what is dominant? I mean, we pick them, those which are informative in Poland, but we base on the, not really looking at the bacteria, but those units. So we look only on the specific sequence abundance. And as I wrote, just about 60% of them are assigned to bacteria. Yeah. So we can check whether they are with this dominant ones, which are defined by a few papers, but we haven't done this. I mean, this is the part of the work which we are doing at the moment. I mean, for this project, we just only look what is informative, not looking into the species really. Now we are investigating what species, what functional species.
Great, we have time for one more quick question.
Cool stuff about these features, basically, how long, what's the size of these microbes, these washes?
The size of the units?
Yeah.
I mean, so here we work because we knew that the probes are 120 bases. So we will limit ourselves to the units which are 200 to 500 bases for the easy design of the probes, but we see way longer. So like for the Horizon one, we are thinking, we start investigating with Andre to taking the Oxford nanopore technology to it until, and then you can design it. But for this, you will need to have longer, units which are informative. Like I think Andre was saying that to make it efficient, the time of read, you need to read about 500 pages, and then before you make a decision, the point should already 5 to 1,000 pages further. Right, so we need to think about, having units of 2 KB or longer, but we check that we have plenty of those as well.
Great, and real quick, because these are soil samples, so the responsible bank was asking that the propensity to find human-associated bugs and bugs is pretty low. So I think, I like the approach, it's really cool. So,
Great, thank you so much.
Thank you.
Our next speaker is Dr. Fumito Murayama, professor from Hiroshima University, who will be presenting on-site experience using suitcase labs. Dr. Murayama focuses on science cultivated within international collaborations. His lab has developed non-invasive methods to uncover dynamics among pathogenic genes in the environment, especially various environmental waters globally. Thank you, Dr. Murayama.
Thank you for the introduction. Good morning, everyone. I'm Fumito from Hiroshima University. Today, before I start, I'd like to thank your committee to invite me to have this opportunity, and I'm really pleased to have a presentation in front of you all. So today, I'd like to explain to you about my laboratory device called suitcase laboratory. That, as you see the title, this is very important for us to have an analysis of sample on site. So, okay, so our laboratory name is microbiology, genomics, and ecology, and the focus is especially aerobiology and also drinking water microbiome. For this research topics, suitcase lab and field analysis is very important. So, have you ever wanted to do microanalysis in tiny area? So, in my image, maybe you know some Japanese animation called Doraemon. So, you know this one, Doctor's Kit in Doraemon, called Doctor's Kit is, as you see in this picture. I can, wait, sorry. Oops. So in this kit, you can analyze the patient's condition, I mean the diagnosis, and also this one includes many devices and medicine inside this small box, like a suitcase. So it's my image of our suitcase laboratory. So this is an image, very famous. Most Japanese people know this image. So in my laboratory cases, I'm also working in Chile, in addition to Japan. Maybe you know that Chile is a Latin American country and the longest city from north to south. The distance is more than 1,000 kilometers. So in my case, I try to analyze the algal bloom in Chile because Chile is very famous in salmon aquaculture. Maybe yesterday you eat a lot of salmon. Main producer of salmon in Japan is Chile. So in the case of Chile, to analyze the harmful algal bloom, they usually take a sample and bring the sample and analyze after 1 week later after collecting samples. It means that if some harmful algal bloom occurs, it's too late and many salmon is killed by harmful algal bloom. Sometimes the damage, economic damage is more than $1 billion US. So to analyze harmful algal bloom on site, I previously developed this kind of small suitcase laboratory. It's already published in 2021. And this suitcase is, as you see, not so big, 15 kilogram. And this includes all the kit and device from sampling to analysis by ramp, like a PCR methods. As you see, there is a bucket. I can show some. maybe here is a bucket for sampling, and then in this case, target is RG. So after collecting water, filtration of RG onto a membrane, and then extract the DNA using simple alkali boiling methods. And then after that, I use the lab methods to identify the RG inside or presence or absence using this simple incubator-like device. So, as you see here, from sampling to DNA extraction and to lab methods, it takes only 2 hours after sampling inside. So this is the example of some field sampling in Chile. And this is just a trial in Japanese using my Honda Fit car. My car is like a laboratory, as you see. So after sampling and the extraction of DNA by Marshall, simple DNA extraction methods, this is useful only for, I think, alkali blue. RG is very easy to destroy and extract DNA. Then I detect the presence of target RG in my car. And this is a Chilean example. So as you see, if I use lab methods, simple detection methods is possible. As you see like this one, if we add the UV LED light, if there is a positive target DNA inside the tube, then we can see the fluorescence compared to negative control. It's very simple. And but now we have this kind of 6 or 8 different non-primer set for targeting harmful viral group. But I think that it's better for us to analyze the bacterial community or microbial community analysis on site also. Because the lab method is very simple and very quick, but as you know, only targeting microbe can be detected. But in our case, as a microbial ecologist, I need to analyze the microbial community on site also. So in that case, unfortunately, the weight is twice double. So it means 30 kilometers. Thank you. Yes, too much. But inside this suitcase laboratory, there is a nanopore MK1C and qPCR and beta beta and cream plus here and HEPA filter. So after the suitcase includes this kind of clean booth inside. This is custom ordered, just fit for the suitcase. So, and in addition to this clean booth, we need to add the silicone out here. And then if we build like this, we confirmed even after 35 cycle of PCR, no negative control band amplification appears, targeting 16x ribosomal universal primer from 8 quad to 1042 reverse primers. So, try to develop this suitcase laboratory take a lot of time. It appears simple, but from real first product one is my real suitcase, like this, very small and simple. And but this brand name is Limoa, but this suitcase is not so good for Chilean transportation. because the Chilean transportation always throw all suitcase and broken only one time travel. So my suitcase is gone now. And then now after that, final latest version is like this, and this is too heavy, but now I can analyze the microbiome community on site. And this one is a prototype and not so sophisticated. And so first thing I did is comparison of PCR prevalence, because the PCR amplification efficiency is totally different depending on the brands. So finally we choose this one. And also we compare PCR machine, because some PCR machine want to amplify very well. For example, this one is very small, but the amplification is too bad. Because usually environmental sample DNA concentration is very low, and we need to select the good amplification machine. And then after that, we try to use a nylon sequencer, because it's, as you know, it's portable. And after our simple informatic treatment, we can identify species level detection very efficiently. In our case, we just use mapping methods to the reference database after just size filtration. For our purpose, it's enough. But more important point for us is condition of PCR amplification. So as you know, maybe some of you have experience of using DGGE methods to analyze bacterial community. Before it's used maybe before coming to NGS sequencing. So when we try to use a quick PCR amplification, the community composition is totally different from original mock community. So in that case, after several trials, we finally use this condition. This one, PCR condition is not so comfortable because I take a long time, take a 3-hour PCR. But after that, the community composition is very similar to MySeq data and also mock original bacterial community structure. So now I'm back to the suitcase laboratory. Why we develop this one? This one I usually use for field sampling, especially for our water microbiome project. So as is known, in Japan, we can drink tap water everywhere without considering to buy water in the vending machine. Maybe some of you know vending machine water in Japan is just like fashion. So you don't need to buy water in Japan. And in our case, we are interested in why Japanese water is so safe. And also, traditionally, many old people especially using the groundwater. So that's why we go to several groundwater, hot spring, hot spring sites, and try to analyze the microbiome in the groundwater. So we designed this kind of protocol. One sample analyzed on site. Another is bring the water in cool logistics, because the Japanese courier is very nice. Because as you know, we can enjoy sashimi or sushi everywhere in Japan. It means that cold temperature logistics transportation is very nice. And just we send the water sample to our laboratory in normal or room temperature, not outside temperature. I mean. So after that, if we bring the water from Hokkaido, for example, to come to my Hiroshima laboratory, take 4 days. Then after that, filters water into 0.2 micrometer per membrane filter and DNA extraction, and then PCR and analysis using the nanopore sequencer, using very, very simple informatics way. And then, as you see, this is a summary of temperature change during the transportation. So this one is called transportation, as you see here, here. So it means usually cold temperature transportation is kept very efficiently and without any problem. And but in the case of outside temperature one, the temperature is around 25 centigrade. It means the temperature for many microbiome. So this is the result of qPCR of 16S liposomal RNA. So this is field real-time analysis of liposomal copy number. And then here is called transportation. This one is after normal room temperature transportation. So here, as you see the summary, there is a big difference between on-site number and after transportation. So as you see, the abundance is more than around 70 times higher in cold one, but in the case of normal outside temperature, maybe more than 100 or more. So it means copy number or biomass totally changed after transportation. Here, 5,000 times, two big difference as you see. And in the case of cold temperature, 7 times in average differ from on-site. So this one is a summary of phylum level analysis of on-site and cold transportation and after normal temperature transportation. So it shows very clearly, as you see, in the case of cold or normal temperature transportation, decreased expect for proteobacteria. So it causes a big difference and biased change in the number and phylum. So if you see the alpha diversity, as you see here, the original one has the highest diversity, but after the normal transportation, diversity totally different like this. So this one is very noisy, but what I want to say is here, depending on sites, the effect of transportation is totally different. Of course, I think that not because of the time of transportation. I believe it might be the reason, the difference of original microbiome structure difference. So this is just a small summary. So proteobacterial function increased in normally, and the percentage of the phylum decreased with after transportation, and number is totally different. So if we try to analyze very more in detail, like genus level analysis, for example, in my laboratory case, we usually very interested in the mycobacterium, because the mycobacterium is opportunistic pathogen of many Japanese old ladies. So, but after transportation of water, even tap water, mycobacterium abundance usually changed and decreased dramatically. So it means, of course, it depends on the target. We must change depending on the target, but we must analyze on site if your target is rare microbe or such as mycobacterium, as in my cases. And then what I want to do next is try to develop a prediction model how the freshwater microbe will change. In my case, I already developed a prediction system used in the case of Chilean aquaculture field cases. So in our case, if we have maybe 2 or 3 years every week sampling data set, we can prepare the mathematical modeling. So in our case, we use empirical dynamic modeling. In that case, if we have a lot of enough data set, we can predict what kind of microbe will appear after 2 hours, 2 weeks, like such kind similar to weather forecast. So just I hope to introduce you about our laboratory other project. So for example, our cases, first, as I said, we have interested in the bioaerosol. So we take a sample from this kind of tower in Fukuoka. And also our campus is located in rural area and a lot of crowd, especially between autumn to early spring, between mountainside, we have a lot of crowd in the morning. So we take a crowd sampling from this kind of tower on the mountain. And also we are collecting a taxi filter to know the normal city microbiome air, and also we take a sample from above a free troposphere. I mean, this kind of light plane we use, this one can go up to 1,500 meters. Not so high, but it's very useful, very easy for us to use. And this one is example of taking a sample in the airplane. We use maybe same as you. We use SASS 3100 sampler with this cap. We usually open this custom cap when we go up to the 1,500 meters high using this small airplane. Usually, because of the low biomes, we usually use 4 air samplers. And maybe this is all from my study today. And if you have interested in our laboratory, please visit to MZE, Microbiogenic Psychology Laboratory, Hiroshima University. This is Chilean hotel, maybe you know. This is image of my laboratory. And finally, I'd like to announce you. I had a seminar last week by Venkat, and also next week I will have a hybrid seminar by Sophia here. Thank you for listening.
Thank you. We have time for questions, about 5 minutes.
Very nice talk, Dr. Mariama. The question is, the data that you have shown are based on 16S amplified thing using as a, the amplicon sequencing using MinION. So do you have any plans to do the shotgun metagenomics in a suitcase lab?
No, I never did that. Yeah, as you know, in our cases, our target is usually low biomass samples. And in my case, as you see, we use MK1C sequencer. It means that sequence volume is not so good for the shotgun sequencing.
That's good. You answered that second question. But finally, the DNA extraction that you are doing on site,
yes,
how good that is when you compare to the lab-based methodology, even though you have a beading there, is there any good for the, you have used for algae for sure, it may be good, but is it good for the spores?
Source?
Spores.
Spore. I see. Yeah, of course, we already tried a lot of different kind of base beaters and DNA extraction kit. So today I just show you the difference in the DNA polymerase and also sequencing artificial conditions. But in addition to that, we also compare the difference in the base beaters. How is the extraction efficiency using the streptococcus species? Maybe some of you know, in all the ISMI journal paper, there is an analysis about the oral the community, but I think definitely is not solved. So we do not know about the oral community. So usually many people know oral community is based, most dominant one is a streptococcus, but the paper don't have only 5% streptococcus. It's really bad because they use the normal DNA extraction kit for E. coli. So it appears no. So I was belonging to the dental department. So I usually use a streptococcus, many different kinds of streptococcus. It's very difficult to extract DNA. So I usually try to repeat the number of base eating time. And then, for example, I add 100 streptococcus, and then after one time is eating 5 minutes, and then the extract DNA, how is the number, and then second time, third time, fourth time, five time, then DNA extract concentration will be stable, plateau. Then I decided how to use a base beaters. So I think the extraction efficiency is very nice because after that, and also I checked the sample onto microscope and then checked if there is a remaining cell or not. So I think almost perfect. But problem in my method is try to destruct everything. So after that, it means DNA fragmentation occurs and not so good for sequence. That is my method's problem.
Thank you for a wonderful talk. One question for you. So that proteobacteria result is really striking. That was really amazing. I'm curious, have you looked yet in these three conditions at viability afterwards? So you have compositional changes, but how does viability change after these shifts?
Viability? Yeah.
Of the community, like how much, if you can per culture X percent in cool versus normal versus on-site.
Yes, very good point, I think. So because in the first slide, I show you what is our laboratory target. Yes, here. So I mean one of our target is the rare microbe. So it means rare microbe is not a whole, but usually, as you know, my meaning is hope to analyze uncultured things, such as phylum CPR and patisserie bacteria, for example. So why I'm doing this kind of analysis in our area is if not today, but I have data in our groundwater in the 16S ribosomal RNA level, but more than 50% of microbe in our groundwater is patisserie bacteria. So it's a very striking and exciting data set for me. And just before coming to here, I again go to the same sampling site and take a sample, and now I try to cultivate the culture the microbe on site because maybe you know this Hiroshima site is very near, but even go to Fukuoka area, I need to drive 5 hours by my car. So it's very good point. And now we are just cultivating what is the difference between on-site cultivation after transportation cultivation. So maybe next time I can show you. Now do it.
We have time for one more quick question.
Is it with swabs, if you culture straight on site, as standard today, the enterobacteria, the proteobacteria, or.
Use the mic, because online people are.
So I thought that was a great question. With the swab samples, if you cultivate directly after you come back from your subway sampling, you will get lower yields of proteobacteria than if you wait a few hours. So my students were always, well, they knew that I'd get mad if they left their samples for too long on the bench. So it's really interesting data discussion here.
Thank you for your comments.
Great, thank you. Our last talk of the morning session is Dr. Katsuri Venkateswaran, who will be presenting Revealing Resilient Microbes Overcoming Metagenomic Challenges in NASA Clean Rooms. As a senior research scientist at the NASA Jet Propulsion Laboratory, and Dr. Venkateswaran's research focus dives into extreme environmental conditions, including deep sea and Mars Odyssey and Mars exploration rovers. He directs microbial environment and sanitation projects, initiatives, and funding using state-of-the-art molecular analysis coupled with nucleic acid and protein-based microarray, with the ultimate goal of setting standards for planetary protection policies. Thank you.
Thank you very much. Thank you very much, Chris, for inviting me to come here. The wonderful place. This is my second birthplace, I can say, because I was here for 11 years. So, konnichiwa for all of you, and who came from all over the world. This is a beautiful place. Enjoy. Anyway, so this is the one that normally I show because the size of the rover since 1996 as a sojourner to the one that is roving in Mars right now is huge. From where we have, some small toy things that you guys might have used it when you were younger, okay? And now it is like a Mini Cooper size. But these things, when exposed to the other planet, should not contain some sort of microbial signatures more than 300 spores per meter square or 10 to the 4 in total. Whether it is a small size like this or the big size like this. So what that means is the contamination level, the requirement, everything is same, but the size increased. It is basically impossible now to clean. So what that means is with the advent of the new technologies like metagenome sequencing and all, we built a database telling that, okay, we carry these hitchhikers with us. So when the sample comes back, you can nullify and telling, oh, these were the ones that taken from Venkat's lab and contaminated Mars. Okay? So I'm actually going to talk there. We have advanced metagenome sequencing and techniques, but what are the challenges? So it depends on people if you are talking. If you are talking to the federal agencies like NASA, they have a requirement to do this kind of stuff. So the requirements is based on some of the microbes that can withstand in such environmental conditions of the space, extraterrestrial conditions, right? So those resilient microbes. Do they get captured by metagenome sequencing? That's the biggest challenge. What are the challenges we are seeing? You may be able to culture them, but you may not see with the metagenome sequencing. So that's what right now NASA is reluctant to put a requirement based on DNA signature. I'm retired from JPL right now, but still I'm having a NASA project and also working with Chris Mason on several different things. The new place right now is University of North Dakota. because Chris did not get me into Cornell yet. So today, I'm going to talk about this, things associated with Mars rover, kind of, assembly clean room. And also, we can exploit the vast nature of the space station realistic. And we take some of the things, resilient microbes, and then put it outside the space station for a few months to years and bring back to see whether they are surviving, if at all, why they are surviving. Is there any useful thing that we can develop from there for the mankind? Okay, so that's what is this. So it is anti-aging cream, a kind of thing, space to your face. So if you want to look young, you can use this cream, which is actually developed from one of our bacteria producing this and being, already in the market. Okay, so the one that I'm always forget because somebody will pull my mic later and I will not be able to acknowledge them. So first I show this always. So many people involved in this in my team, and some of them are here. Karthik Drew from India, and a couple of students are going to have their presentation, Pratyay and Shoman. Thanks for Chris to support them as well. And I have a couple of mafia. They are with me. One is Chris, and the other one is Nikos. So they are with me in my thing. And recent one is Pavel is coming in to mine. And still this mafia group is going great, and we are hungry. Okay, even though that I'm retired, but I'm still hungry to do a lot of science here. So with any of you that we can do it. Having said that, what I'm going to talk about it. What the hell NASA is doing with microbes? These are, NASA means engineering, right? But think about this. You want to know, the biggest mission is, are we alone, right? Are we alone in this planet? Did we come from somewhere? If you want to go on and look for those kind of signatures, then don't contaminate the other planet. That's called planet protection comes. But by simply restricting yourself, by putting such a kind of thing, it's not going to work. So we have to move forward with human habitation in other planets. For that, you cannot take everything with you. Can you use the microbes as a resource? That is called space biology. Can we use microbes and then see whether how they can manage such conditions and produce certain kind of different products of human, for agriculture? Can we produce some sort of consortium that helps these microbes grow, plant growth in other planet? And human research program. The benign microbe, that human associated skin microbe like Staph aureus, because of the condition that it faces in other planet, whether it is going to shed some of its mobile element into a normal bacillus, and that can survive in radiation conditions and become problematic for the human. So because of that, microbiology is very important in our institution. So here I'm going to talk about more in detail about rare culturable microbiome. Can we detect or miss them with the state-of-the-art metagenomics? That's one thing. Why do we need to do that? Because culturomics in space science is such a translation science, and its benefits is very, very important for tomorrow's human exploration in other planets. So antimicrobial resistance, which I already told you, the human are going to be there in an enclosed place, so confine the area, so mobility is less, and you're always be stressed under the immunocompromised conditions, so the microbes may attack. Like, think about ICU unit, okay, where the patient is not moving, but microbes may attack them more easily because you are immunocompromised. Same thing the astronauts. Astronauts are very well healthy individuals, right? Over the period of time, if you make them like COVID time, how our mental things are different, right? So same thing. So AMR will be different there. But also the other thing is, one microbe may produce certain things, and act upon other things. So microbial warfare, how the community is doing. So for that, you need some culturomics to do. And then prevalence of AMR on space station surfaces will help us to understand what needs to be targeted, what kind of cleaning reagents you can have to remove them. And these strategies might generate some mutations or some modifications that they have to adapt, and in this case, it produce some biotechnologically important compound that I wish to tell an example. Here is a video that I will show you about what is the Mars 2020 rover is doing, how it is drilling things and other things. So this completely robotic mission, as you all know. It's a rover roving around. First, take its own camera and look for where you need to go and locate, collect the sample. Okay. Now the drilling component come out and then put it stand on top of the place. Oh, this is the correct place that I would like to get because of the radar and other things. It's going to help us to find a suitable place. Now it is going to drill about not a mile down, but it's just a few centimeters right now. We put about those kind of things are collected and put it in a sample cache and bring it back later. We do not know how to bring back now. We know how to send it. We do not know how to come back. We are still developing. For that, you cannot simply keep quiet. It may take 10, 12 years. So maybe in 2035, we'll get the samples back. Until then, we need to know how to keep this clean and kept it in a clean place. So this sample container baked to 500 degrees centigrade. Okay, and it is completely sterilized that way. And right now we are measuring the volume, sealing the sample tube, image at each step, move tubes to storage. Then, when we know how to bring it, we will bring those samples. Until then, it is going to sit in Mars. Okay, so now you know why we need to do some sort of microbiology in other planet, right? So this is what we do normally. This is the video telling you what we are doing.
It's really important that we send a rover that's clean and we make sure that it doesn't contaminate Mars. My name is Lucy Stricker and I protect Mars from Earth bacteria. The next Mars rover is slated to go to Mars, collect samples so that eventually we can bring those samples back to Earth and determine for the very first time, did life exist on Mars? There's nothing that we can build that's sterile. So we take swabs and life to the spacecraft as it's being built. It gets put in an oven, it gets put in various chambers and clean rooms so that we can maintain that level of cleanliness. If we do find something on Mars, we have to make sure it's something that actually came from Mars and not something that hitched a ride. This is the place where the magic happens. Oh, that's really cool. In this lab, we look specifically at spores. So spores are those tiny microorganisms that can actually survive if it made it on the spacecraft in the journey through space, through the vacuum. It's very humbling to be a part of this big project because there are hundreds of people that have to come together and build a spacecraft. There's no one person that can say, I did this, I made this happen. It's always a we. I owe it all to Carl Sagan and watching the cosmos. I remember being a little kid, going to the public library, renting that VHS. And from that moment, the light bulb turned on. It actually was the start of my passion of science communication. We are citizens of our universe. We have to be good ambassadors when we are exploring other planets, other moons. So it's the right thing to do.
So there is a
It's
thing
really important.
There is a thing we normally do. Okay, so she was my postdoc, and we hired her as a scientist, and she became my manager. So. All the young people, please think about that. You can think big and become someday, you can be a boss of your PI. Anyway, so this is the topic, rare microbiome in metagenomic approach, right? So the metagenome sequencing in NASA mission critical clean room environment encountered challenges in detecting extremophiles due to their resilience and low abundance. Guess what? You have a million read. you will be putting what is the top 10, or whether it is abundant more than 1%. The rest you just throw away, or put it unclassified, or put it others or something. But that is what I want to know. That's what NASA wants to know, because that's where you have the best that might survive the other climate conditions. That's what I wanted to throw the challenge to this community, how to get rid of the dominant microbe that is normal, and how we can look into those one that is abnormal. Okay, so stringent decontamination measures and nutrient scarcity, which is important, oligotroph, okay, resulted in less metabolically active extremophiles. That's what I'm trying to work with Nicholas about, how to get these things where, these extremophiles are less active metabolically, but need a co-dependent to do it, okay? But this we right now is underrepresenting in shotgun metagenome sequencing data. Why that is, we do not know. The robust nature of extremophiles, particularly spores that is based on NASA requirements right now, protected their DNA, complicating extraction and potentially compromising genetic material quality. So we need to have an appropriate thing that can break open the spores, but also the benign microbes should not get chewed up. Okay? So the genetic material was often overshadowed by more abundant microbes, complicating the detection. So what are the limitations, which I already touched something? So comprehensive reference database. We cannot simply say it is not there, unclassified. We need to know, so the functional whether that is doing something or not, right? The other one is continuous database update. Without that, metagenome is going to tell you that unclassified, that is not there, or it may be a microbially dark matter. Contamination by even low abundance microorganisms can have serious implications for NASA. Okay, so these are the ones that we need to work on. So maybe targeted enrichment techniques and advanced DNA extraction, that's what what I was talking with Howell, that if we are able to target this spore specific thing, that may be good. Then we can make a requirement for NASA. So culturomics and metagenomics, right? So, over 15 plus years, 2,500 strains we have isolated, and with the help of Chris Mason, we have actually sequenced all of them, but that shows 100 plus novel species. Having but just having a novel species, naming it, we are not Baptists. I don't want to be that kind of person to name a thing for the sake of it. But it has to show some functionality. Why do we need to do that? For example, we did Acinetobacter pti, which is actually not a big, AMR thing, but in space station, it become actually acquiring a more antimicrobial resistance that become a problematic like Acinetobacter pneumoniae. So those are the things that we have to do, and the sequencing culturable extremophiles might aid us to get some sort of protocols to get it off. So for all those things, you need a culturomics. Metagenomics from the clean room are also investigated. It's not, we are not leaving that, but we want to know what is the percent of these novel microbes in metagenomic signature. That makes sense, right? Let me give you an example. So this is a clean room where we are building some spacecraft, in this case Phoenix, okay? And then we get the DNA, we get the isolate first. We grow them, culturomics. I think Brandon was asking whether you are also doing culturomics. So we do culture-based analysis, then the DNA extraction, whole genome sequencing, then after all these things, quality control, blah, blah, kind of thing. Then final assembly comes and the genome-based indices will tell you whether novel species or not. In this case, 200 out of 215 strains, 53 were novel, that is almost 25%. If you compare that to your garden soil on your backyard, it is only 1 to 2% novel species you will find, because You have so much abundant microbes out there that mask their cultural techniques. But in this case, you already clean it, the dominant microbes are get cleaned. So you have the tail of those things, the 10% of what you normally see is still there, so you will be able to find more, novel species. So we also extract the DNA of the same samples, split into culturomics as well as metagenomics, and then do with and without, PMA treatment, which tells you whether it is viable or not, even DNA techniques. And after that, the raw reads and do whatever you guys normally do, and then see, presence and abundance of the novel species in the clean room via metagenomics. So this is an example I'm going to show you based on this Phoenix data. So this is shotgun. If you use the shotgun, you'll get all this kind of nice tree, I call tree. So this is comparing ISS, JPL, and Kennedy Space Center. The red dot, green dot, and violet color is 3 different places. Okay. If you put the human like space station, obviously you will find this kind of bacillus and other things come in there. Okay, but the same thing are not seen in the JPL and KSC, which makes more important for NASA. NASA wants to see what are the hitchhikers that can be present on the surfaces at the same time resilient to these extreme conditions, right? But in space station, that's okay. But if you are taking the similar thing to the other places, it should not be, okay? But it's masked, that's why. But if you culture them, you see them, whole 9 yards, including half of them are spore formers, other half are not. Okay? So these are the 26 novel species that I was talking about. Okay? So now, these are the phylogenetic tree, where it is placed and all, but the take-home message is based on whole genome sequencing. 215 isolated from the Kennedy Space Center were transferred into 3 phyla. 25% of these strains were now, and most of them belongs to bacillus. You are getting those are the ones that survive in other planet. It will be missed if you're using it, next generation sequence only. This we also do those kind of stuff, and this is important. So now you have metagenome data that conducted in 2016 and 2018 also to see whether these novel 26 species, how this persists. Very, very, very low bar. In some cases, there is a 1 or 2, but what I'm trying to say is by average, the coverage breadth range from 0.00045 to 4% in JPL SAF during 2018, and 0.004% to 6.8% in Kennedy Space Center. So in metagenome, what we do, less than 1% will go into others. So we don't cover this kind of thing, which are important. In the interest of some time, I'm going to skip a few things because I have to connect why do we need to do this, right? Culturomics, right? So one is to see whether these are problematic things for other planet. The other one is because astrobiology get compromised, you are sending $2 billion worth of a mission, come with your own contaminant, it's a problem, right? But now, at the same time, can we use that translational, is there any translational benefit for the mother yet? Okay? So from basic science to enhancing human health in a closed system is one of the things. The pathogen detection and countermeasure development, and leveraging this omics data sets for medical practice. So I'm going to, I have 4 different applications, but in the interest of time, I'm going to stick with this item number 4, which is bacillus suffer 32 story on space to your face thing. Okay, genomics, proteomics, metabolomics, everything done based on that. We have found out of the world space to your face product out. Okay, so this is, as I said, you have a space station, you have a big real estate, you are exposing outside the space station, and first, you cannot simply say, hey, I want to take this bug, because class love this. Okay, you cannot say that, and you have to show why. So initially, 1999, I isolated this bug, and after that, a couple of years, in 2002, we sequenced one bacteria, $100,000, 100,000, okay? So now you are doing what, $100, right? So there is before NIH came into that human microbiome, so we were able to get this whole genome sequence. And then we also showed more simulation studies on the earth. I'm going to show you again. And then found these are the one better that we know today in terms of the resistance, whether it is chemical or physical. Okay, first, let's see. Normally, you guys are all using the UBC254 as a biocidal agent, okay? So you see that Bacillus subtle is 168. That is the American Public Health Association is coming up. If you have water, that requirement is 200 joules per meter square, you kill everything. That's what the standard right now is. So that's what the drinking water and other things that we are drinking. But there are certain microbes, Bacillus pumilus, ADCC27142 can go to 1,000. But our little champ, the pumilus Sapphire 32, only 1 log reduction even after 2,000 joules. Okay? Now, that's not the case, but Martian UV is 200 to 400 nanometer. So let's see how it is. First, you have to have an appropriate setup. Here is the sample holders, where you have 30 millimeter size of the metal coupon. You can dry the coupon, dry the spores, you can put it on it, and then put all the sensors together, okay, and then vacuum, because 6 millibar is Martian atmosphere, okay? Give it the vacuum by pushing into some sort of, gases, and also vacuum. Then you have a chamber that has this 200 nanometer, 200 to 400 nanometer UV setup there, solar thing. This is a glow bug that you can use to check whether your bug is surviving or not. Yes, it survived. See, in 15 to 20 seconds, that Bacillus subtilis 168 can get killed. And even that Bacillus megatherium ADCC 14581 can be killed in 5 minutes, but you have only 3 log reduction with Bacillus pumilus 30 minutes. Somebody may say, hey, let us wait 30 minutes before we explore things, but it is not the case. You saw the wheels that is rolling, that cannot be completely exposed to the UV, Martian UV. So that means when it roams around, when it goes to the Martian soil, it might get transferred into the soil. Once it is having a shadow, it can happily live there ever, millions of years. But yesterday there was an announcement that NASA has said that Mars had water. So if you drill down 1 mile, you will be seeing the water there. Whether it is bioavailable or not, still we do not know, but water has been found. Now, with that kind of thing, if you have this spores mixed with the soil, even for 1 day of Martian UV will not kill. Okay, now you have this data, simulation data, everything. You work with the NASA and then say, hey, I want to take it to the space and then expose it outside. This is the space payload. Each, the hole that you're seeing is 13 millimeter. You can put your stuff and then do the, Martian, space UV, that one. The other one is Martian UV. And you can also close to say, okay, there is no UV passing through. Yes, after coming back, it went in 2008 STS crew, and we mounted on the outside this, Columbus module. This is the ESA, NASA project, and you see those things that is, I'm not able to show it here. So this is what it is, and then here it is, and here is our stuff that is exposed for how long? 18 months. Okay, 18 months exposure in real space condition, then brought back. And then when we look into those things, this is after 1.5 years, This is the bad aluminum, this is the monolayer. At some places, then with the metal, it can go in there and get shattered. So it can live, but it may not proliferate, but it can survive. That's a key point for the spores. So after coming back 1.5 years, you are seeing the shape intact. These are all environmental same. You are not removing it and doing anything. Take this thing and put it on environmental SEM. And so we published multiple papers on this, and we didn't see genomically any difference. We have done multiple times. Genomically, we don't see any difference because it is a spore. It will not change anything, right? But maybe having a protective layer that is absorbing the UV, metabolically different, right? So this is the spores after coming back, the UV space conditions is having twice the resistance compared to the control, which I already showed you in 2000 joules, you might die off, but in the one that returned back, the UV space, it's not even up to 4000 joules. So, spores again is metabolically inactive, but you need some vegetative cells so that you know what kind of metabolites you can look for. So, vegetative cells is unheard of 900 joules that you can make them live, but normally 200 joules, including the control that did not fly, was kept here, die off in 200 joules. So, proteomic characteristics is telling us that there is something different here that we can exploit. So we did so many different things. So taken together, this functional assays demonstrated that the activities of TCA cycles and energy metabolism were decreased in space surviving strengths. So that's what comes to the metabolomics, the clinical trial, everything. The simple lysate is telling that it is actually producing some age-defying serum that's good for the young girls who wants to be looking young even after 20 years of MetaSUB meeting. They can use this age-defying serum, and there's nothing but it is having all these things. It's nothing but hyaluronic acid, but the hyaluronic acid produced by this space-flown thing has better activity than the chemicals that they are using it normally in these kind of foods. So with that, this one, because of the time that I already told you all this stuff, this is based on 25 years worth of 20 different postdocs, and then 100 different students who are working with me and with the countless collaborators, including people like Chris and Nikos and others. Thank you very much.
Great talk. I actually have a quick question as a fellow space nerd. So I was wondering, in terms of biologically interdependent microbes like nitrogen fixers, other than maybe bacillus, have there been any uncovered or new resilient species within the Martian environment and the atmosphere that have potential to have that?
Correct. That's a very great question. That's what I said, loosely attached to my thing. Why do we need to do this resilient thing, which also have some potential as producing plant promoting growth hormone, which we also found and then published. The Sphingomona species, and that has an Methylobacterium species. These are all novel, but still that we found through the functional characteristics, they produce some of these growth promoting hormones that we mixed to show how it can grow under the space condition. That's a whole entirely different topic.
That's really interesting. Thank you.
Yes, sir.
Great talk. I can see now why you're so resilient to retirement. So it makes a lot of sense, of course, to apply sterility checks in the NASA clean rooms, because that's what they want to get rid of all microbes from there, right? But inadvertently, you have ended up coming up with a mechanism to enrich the rare biosphere. Have you thought about applying the same approach for any other habitat? Soil, marine, fresh water, whatever, in order to be able to give the chance in the rare biosphere to be cultivated. And that's the other interesting part of that.
Sure.
A big part of that is cultivable.
That's right.
Cultivatable.
That's absolutely right. So, one thing is, people tell NASA, hey, you are making superbug. Why? Then we cannot build a spacecraft on our garage. That means that we're contaminating a lot of things in other planets. We do not know which we are taking. So we have to take, to some extent, this kind of thing. So that is one thing. But still, we want you guys to help me to get rid of the dominant microbes from the metagenomic sequences. Look for the one that is less than 1%, how that is actually shaping and run. Not only clean room, but also soil, thing, So we simply say matter will dark matter, this and that kind of thing, mainly because we don't have a database. If you put 100 different kinds of, culture-based analysis or something, you might be getting those red flags. I think there is a talk today, during this meeting about cultural mix, how they are going to try to increase. So I would like to get those information from you guys, and I'm interested to see, and also with Powell about the targeted, sequencing approach of the spores. We don't have a spore-specific gene that you can bring it up. It may be UVR gene. Then Deinococcus will also pop up, or something that gets the UVR gene will also pop up. Is that right? So we need to come up with something that is unique to spore, so that NASA can come up with a requirement. That requirement means you need to have some numbers. Go ahead, Klaus.
Thanks for the good talk. So basically now, when it comes to the enrichment technique, I was curious about that, because I didn't really get much detail. So classically for bacilli, one actually slaughters the population, and then induces germination is one way to get usually a germination game. So I'm curious if you guys have that as a control, and then one can always incorporate some of the methods used for the extremophiles or the archaea, where one actually carried out culture in stepwise increments of temperature, changes in sulfur concentration, et cetera, et cetera. I wondered if you guys had tried one of these more massive, I would say, conditional.
Great question. There's no question about it. Okay, I'll answer one at a time. So we are take the sample, heat shock them first. So the one that comes back, the heat shock tolerant microbes, can grow in other regular media. Okay, then you will get vessels. But the one spore formers that can withstand cyclophile, the one that withstand alkaline conditions, the one that withstand so many other functional things, cannot probably live. So how many culture medium I can go? That's why NASA is interested to look for, get everything, and then look for the signature specific for spore forming microbes, whether it is bacillus, or alkali bacillus, or lysine bacillus, all this kind of thing that we have right now. And also the spores the community of producing fungi, it's also there. That can also withstand in another planet. And coming back to the archaea, yes, we have been extra, we are so cautious, and then did so many work since 2000. The archaea is more or less than virus in this clean environment. Somehow we are cleaning to the extent level, but if you're amplifying archaea only, you may be able to get it. But that's not the case. We need to come up with some requirement, right? We can clean to the level, right now is 300 spores per meter square. Otherwise, it will be humanly impossible and no mission will fly. So one of the things is archaea, we are interested because there's another thing, those archaea may withstand other conditions, other extraterrestrial conditions, but very, very low in clean room.
So I have a question right here. So it seems that we are standardizing and benchmarking our web app methods and lab computational methods to detect things that we know pretty well, like DNA, RNA, certain molecules. So I'm wondering is NASA trying to control or thinking about other potential molecules that might be a little bit different than the ones we're standardizing for?
Control what?
Other molecules, like other type of certain
Right now,
proteins or things that are not here.
right now we are struggling with DNA and RNA because of the low biomass. You know? But further down, protein-based analysis and other things, may not be possible. The one that I'm showing you are clean room environment. I'm not showing you on the surface of the probe itself because we are not allowed to touch the surface of this probe right now because the requirement says don't go and touch, and then you are contaminating that more than what is out there. So we are doing a proxy that clean room environment may be 100 times more contaminated. Still, we are using that as a kind of test bed to develop technology for the spacecraft surface. Okay, so protein and other things. If something comes better, definitely we can suggest. Right now, because of the DNA era, shotgun metagenomics and 6 and other things that we had done before, so based on 6 we could not come up with the requirement, but maybe based on the metagenomic, we need to come up with something of some quantitative metagenomics, otherwise it will not fly. Without the quantitation, the requirement cannot be placed. For the federal government, right? To become the policy, it should be quantitative metagenomics. If anybody interested on quantitative metagenomics, then we can talk about it. For low biomass, not my saliva.
I'm sure a lot of people have more questions for all the speakers this morning, but we are wrapping up for the morning session, and there's coffee outside. We have time to work together and talk about all these things. Thank you. We're reconvening at 2 p.m.
Thanks, everyone. As just a reminder, if you are going to go pick up food, that please don't eat it inside the venue hall here. There is a cafe downstairs as part of the museum that you can go get food if you'd like there. There's also more options in the agenda booklet at the end for other food options. We invite you guys to go out, speak to the other attendees here and collaborate. And then we ask that, as Marissa said, everyone return by 2 p.m., when we will proceed with the afternoon session. If you have any questions and speakers in the afternoon, please feel free to check your slides for this afternoon as well. There's coffee outside.